Welcome to Study Loop.
So this question deals with the trip operon. The trip operon is the DNA sequence that governs whether the amino acid tryptophan is synthesized or not. Bacteria like E. coli can synthesize tryptophan on their own if it cannot ingest it from its surroundings. So this is a sequence of DNA that you turn on if you don't have tryptophan. So in the two instances that I've drawn here, on the left is the operon in its off state because the repressor, drawn in green, is bound to the operator. On the right-hand side, the operon is on, the repressor is not bound, and the RNA polymerase is bound and can then transcribe the gene. So in which one of these instances would you expect for there to be tryptophan? Well, if you have tryptophan, you don't need to make it. So the left-hand side would be the case where tryptophan is actually present. And the repressor is actually activated by the binding of tryptophan. So when tryptophan binds to the repressor can then bind to the operator, and that prevents the synthesizing of new tryptophan from the cell in itself. So if there's a mutation that prevents the repressor from binding to the operator, well, then you have a case where your operator is always on because the repressor will not bind. So you're constantly making tryptophan, whether whether you need to or not. So let's look at our answer choices here. Choice A, in the absence of tryptophan, genes will not be transcribed. That's not true. We just established that they will always be transcribed, whether there is tryptophan or not. In the absence of tryptophan, only genes TRPE and TRPD will be transcribed. That's not true. All of them will be transcribed. And by the way, when they're talking about TRPE, TRPA, so this right here is actually divided into little sections, and each one of these can make a different protein, all of which perform a different step in the synthesis of tryptophan, because tryptophan is a complicated molecule, so you need several steps to make it. So each one of these guys performs one of those steps, and if you need to make tryptophan, you need to activate all of them, right? So if you're going to turn the operand on, they're either all going to be synthesized or none of them are going to be synthesized. So you don't have the selectivity. Choice C, in the presence of tryptophan, the genes A through E will be transcribed. Yes, that is true. So even when tryptophan is there, they will all be transcribed. So C is correct. And choice D is another one of those instances where it's selective, and that's not the case. So C is the correct answer.